Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Today's topic is expression vectors, features and types. So in this you will be learning about what are expression vectors and what are the different features of expression vectors and different types of expression vectors, and you will be even learning about the prokaryotic expression system and eukaryotic expression system in brief. Dear learners, let's study about the expression vectors in detail. So these expression vectors are cloning vehicles which carry our desired gene and also allow the expression of our desired gene. So to promote the gene expression, these vectors contain additional elements called as regulatory elements in its genome. So if you see these particular expression vectors, they are used in the production of transgenic organisms and used in the treatment of this gene therapy. So if you see the structure of the expression vector, it will be looking like this. So it will be containing all the elements similar to that of the cloning vector, that is origin of replication site, markers, selectable markers, then polylinker site. Like that, these are the similar features to that of the cloning vector. The additional segments which are present in this expression vector are the regulatory elements, like this promoter, which promotes the transcription of our desired gene, the operator which regulates the gene expression, then this particular repressor, the gene encoding repressor, it will be also be controlling the gene expression. Then here you will be having a ribosomal binding site. So here from this is the site from which the translation process it is initiated. Like this, the genes which are necessary for the transcription and translation of the desired gene are present in the expression vector. Okay, so now coming to the features, let's study all the elements which are present in the expression vector in detail. So the first one is the origin of replication. You know that this replication replicon, it is used to multiply the genome independently or autonomously. So the vector, it will be multiplying, and along with that the gene which you are inserting, it gets multiplied when the replicon is of relaxed in nature. And the small size, the vector should be of small size so that you can easily manipulate. The entry and the recovery of this vector should be easy. So that is, it must enter into the host cell easily, and also the recovery from the host cell, it should be hassle free. Then the next one is the polylinker site or multiple cloning site. So these sites, they contain several unique restriction enzyme cleaving sites. They are clustered in one location, and if these, let's see that if the this restriction sites, or if they are present here and there, so what happens means the vector is broken down into fragments. If they are present in one location, they are clustered in one location, you can just cut the vector only at that site, and you can open the vector and insert our foreign gene. So that is the use of polylinker site. Then the selectable markers. So in order to identify our transform cells, you will be using selectable marker genes. The most common marker is the antibiotic resistant gene, and the thing which is present particularly in this expression vector is a scoriable marker genes are also present. That is a GFP gene, GUS gene. These are the reporter genes which are inserted in the expression vector in order to know the gene expression, whether the inserted gene is expressing or not, and how much it is expressing. That is, you can quantify the amount of protein produced by the expression vector using this particular reporter gene. These reporter genes are exclusively, they are present in this expression vector. Next, the the main elements or the regulatory elements which are present in this expression vector is the promoter. So this one is found upstream to that of the gene insert, that is to the target gene. Near the target gene this promoter is present. So this site, it helps in initiation of the transcription. When the RNA polymerase enzyme, it goes and binds to the promoter, then the transcription process, it starts. Particularly this promoter region, it contains two conserved or consensus sequences in the -10 and -35 region. That is, -10 means that is the transcription start point is represented as +1. So 10 base pairs away from this transcription start site is called as -10, and 35 base pairs away from the transcription start site is called as -35 region. So this is the promoter region where the RNA polymerase enzyme, it will be binding. So these are the consensus sequence or highly conserved sequences. In prokaryotes, it is the Pribnow box or the TOTA box, because it is containing TA, AT sequence. It's also called as TOTA box, and the -35 region is containing this 6 base pair sequence TT, GA, CG. So these regions, at this particular site, the RNA polymerase enzyme will be coming and binding and initiating the transcription process. So there are two types of promoter: constitutive promoter and inducible promoter. So here a constitutive promoter, the promoter is unregulated, and you will see continuous transcription of our associated gene, so which is not desired. So that is constant expression, gene expression is seen. So when this particular property, it causes the production of excess protein, which is toxic to the cell. So this feature is not desirable for an expression system. The constitutive promoter example, it includes gap promoter, which is encoding the gene glyceroldehyde 3-phosphate dehydrogenase. Next, coming to the inducible promoter. So an inducible promoter is the one which works in a regulatory manner, and you can control the gene expression. So whenever you want a protein, if you it will be having this switch on and switch off mechanism. By just using certain molecules, you can induce the gene expression, or you can repress the gene expression. So such type of promoters are called as inducible promoters, and these are the promoters which are preferred in the expression systems. The inducible promoter examples, they include this AOX1, GAL1, GAL10 and LAC-UV promoter. In this example, we will see how the gene expression is regulated by inducible promoters. Here you are seeing the LAC-I. LAC-I, it encodes a repressor protein. This repressor, when it goes and binds to the operator region which is present next to the promoter, so it stops the initiation of transcription. When you add an inducer called as IPTG, IPTG is a lactose analog. So the repressor which is produced by this LAC-I, it binds with the inducer, and this complex is unable to bind to this operator region. So once this operator region is free, then on the promoter, the RNA polymerase enzyme, it comes and binds and it initiates the transcription of our cloned gene. So in this way, the LAC-UV promoter is an inducible promoter. The transcription is initiated by the inducer called as IPTG, and the transcription can be repressed or stopped by means of the only the repressor molecule. So in this way, the gene expression is regulated in expression vectors. Next, coming to the other feature, that is the ribosomal binding site, and which it's containing the translation initiation site. So this is the site where the ribosome comes and binds and initiates the translation process. So this ribosomal binding site, it contains translation initiation site. In case of the prokaryotes, it is the Shine-Dalgarno sequence. In case of the eukaryotes, the translation initiation site, it contains Kozak sequences. So in this picture you will be seeing how the ribosome is coming and binding to the mRNA. So this is the translation initiation site of a prokaryote. So it is containing a Shine-Dalgarno sequence, that is the 8 base pair sequence. It is highly conserved. So this is the portion where the ribosome it comes and binds and it initiates the translation process. So next feature is poly A tail. The poly A tail or the polyadenylation tail is present at the end of the mRNA. So this is formed by the addition of adenosine nucleotides at the 3' end of the mRNA. So presence of this poly A tail, it gives a stability to the mRNA and it protects the degradation of the mRNA from exo and endonucleases. Next, coming to the other feature, fusion tag. It is also called as affinity tag or purification tag. So this is a short stretch of DNA which is attached to our desired gene, either at the N terminal or C terminal region of that desired gene. This affinity tag is attached. So this tag, it produces a small peptide which is present along with our recombinant protein. So the small peptide plus our recombinant protein together, it is called as fusion protein. So these fusion proteins, they are stable in nature and they are resistant to proteolytic degradation and they are more soluble. So the presence of this tag, it helps in the easy purification of this particular recombinant protein from the cell lysate, and also it helps that is whenever you are adding this that is the total mixture containing the host proteins and our recombinant protein in the affinity column. So the tagged proteins, it attach to this affinity column and they can be easily separated. The most commonly used tags in the expression vector are His tag, GST tag or MBP tag. Next, coming to the that is the here it is the picture shows the overall elements which are present in the expression vector. So these are the regulatory elements, that is the promoter, operator. So promoter is the place where the this RNA polymerase enzyme it comes and binds. Operator, to this operator the repressor, it will be binding and it stops the transcription or it regulates the transcription process. And the RBS is the ribosome binding site where the ribosome it comes and binds. Then this TAC and ACT, this is a transcription start point and transcription end point, and the portion between this start and end point is our structural gene open reading frame. This is the one which is actually it is transcribed and made into protein. And so once the structural gene is undergoing transcription, you are getting an mRNA which is having a 3' and there is a 5' and 3' untranslated regions, and here you are seeing the initiation codon is AUG and the stop codon UGA. So this one is after the translation you are getting a protein having an amino terminal and a carboxy terminal end. Okay. Now let's see about the examples of expression vector. Here you are studying about the PET vector. P means plasmid, E means expressing, which is expressing through T7 promoter. T means T7 promoter. This vector is having T7 phase promoter, bacteriophase T7 promoter. Okay, so if you see the structure of this PET vector, so the size is nearly 5.4 kb. It is having ampicillin resistant marker gene called E1 origin of replication. Then also it is having a F1 origin of replication. It allows the that is the production of single stranded DNA molecules. When it is infected with M13 helper phase, you will be getting a single stranded DNA. And apart from that, it is having the regulatory elements, this LAC-I and LAC-O. Then P-link means polylinker site. This is the polylinker site where the vector is cleaved with the restriction enzyme, and then the desired gene is inserted. So the promoter here is the bacteriophage T7 promoter. It's a very strong promoter. It helps in the synthesis of our desired gene protein. So that is the main thing. This lac operator region, it is regulated by means of the inducer IPTG. Okay. Next, coming to the other example, PTRC His vector. TRC is a hybrid promoter containing a TRP promoter plus LAC promoter, and the His means the presence of histidine tag. The fusion tag here is the histidine, so it is called as P means plasmid. PTRC His vector. So this vector is of size 4.4 kb. It is having ampicillin resistant marker gene, origin of replication derived from Col-E1 plasmid, and the mutated this LAC-I repressor gene. So it is a mutated one, produces LAC repressor, more LAC repressor protein than the normal one. LAC-IQ is a mutated form of LAC-I. Then you are having this LAC-O operator. Then this histidine tag, it is the fusion tag, and then apart from this you are having this EK site. EK site is the one, it is having the recognition sequence for enterokinase enzyme. So this is the recognition sequence specific for this. EK means enterokinase enzyme, and polylinker, P-link means polylinker site. At this polylinker you will be cutting the vector and inserting our desired gene. Okay, so if you see this affinity tag, it is the histidine tag. So this due to this tag you can easily purify the fused proteins by affinity chromatography, and this enterokinase, whenever you add enterokinase enzyme after the production of this fusion protein is containing the tag plus this XXX, it represents our desired protein, and in this manner you will be getting the product, the fusion protein product at the end of the gene expression. So whenever you add enterokinase enzyme, it will be cleaving at this particular site, that is after lysine. The enterokinase is having this recognition site. The red color one is the recognition site of the enterokinase enzyme. So it will be cutting that protein at the lysine. After lysine, it will be producing the cut and releasing our recombinant protein. In this way, the tag is removed from the fusion protein by using the enterokinase enzyme. So up to now you have seen the examples of these expression vectors. These expression vectors, they require a specific host cell to have the protein expression. So whenever you introduce these vectors in the host cell, then only the gene will be expressing and you will be getting a protein. So here you are having two types of expression systems. One is the prokaryotic expression system. That is, if you are introducing the expression vector in a prokaryotic cell, it is called as prokaryotic expression system. The examples, they include E. coli expression system, and the second one is eukaryotic expression system. So here you are introducing the expression vector in eukaryotic cells like yeast cells and baculovirus infected insect cells or mammalian cells. So such type of eukaryotic cells, if you are introducing, it is called as eukaryotic expression system. Let's have a brief outlook on the E. coli expression system. The E. coli cell which is taken here is a mutant strain. The genome of this E. coli is modified, and this type of E. coli cell, it is taken whenever you are using PET expression vectors. You know that PET expression vectors, they are having T7 promoters. So for that reason, these E. coli strains are taken. So what are the changes seen in this mutated E. coli cell means two changes. First one is this particular genome is having the RNA polymerase gene which is taken from T7 bacteriophage. So now the host cell is having the ability to produce the T7 RNA polymerase. So when you introduce the PET vector, because they are having T7 promoter, and by taking this RNA polymerase from the host cell, it will be able to express our desired gene. So that is one change. And the next one is this particular strain is having the ability to eliminate the action of host proteases. The proteases present in this particular cell, they are inactive. So when these proteases are inactive, our recombinant protein is safe and it is stable. So this is the mutated strain of the E. coli, which is having inactive proteases. The host proteases are inactive in nature. So these are the main features of this that is the genetically engineered host E. coli BLDE3 strain, which is taken for the expression of these PET vectors. Next, coming to the conclusion, so let's recap the main points. So expression vectors are those which are used to express our desired foreign gene, and in the features of the expression vectors we have seen that these particular vectors, they contain regulatory genes like promoters, operators, terminators, then ribosomal binding sites, fusion tags for the effective expression of our desired gene. In the examples of the expression vectors, we have learned about two vectors, PET vectors and pTRC His vectors. And in the types of the expression system, we have also learnt about the prokaryotic and eukaryotic expression system. And in the prokaryotic expression system, we have seen in brief about the E. coli expression system. And with this, I conclude the session.
Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.